Good afternoon. It's great to be here. It's nice to follow on such interesting and relevant talks today. I hope to kind of pull up some of the themes that we've been discussing and apply them to the context of challenge studies to address COVID-19. And ultimately, the take-away message of my talk will be that it's important to invest in these studies, but we can't yet greenlight them. I will say it's nice to follow on from the last talk, because one of the key issues that I think is important to recognize for these studies is there's this need for coordination. And we're starting to see unprecedented coordination in the conduct of clinical trials, but we need more of it to make these studies an option that's worth pursuing. So to give you some background, if you're not familiar with controlled human infection studies, they're sort of shocking to hear about the first time that you do. They're also called human challenge trials, human infection challenge studies, voluntary infection studies. There are a number of names that people have used, but the one I think that is most common right now is controlled human infection studies. And the idea behind these studies is in some ways simple. It's to take a volunteer and deliberately expose them to an infection or pathogen to rapidly and carefully test a vaccine or a drug, or to find out about the disease in ways that would be very difficult to find out otherwise. So this man who's smiling at the camera is a cholera challenge study participant from the University of Maryland. And you'll see he has the number 26 on his whiteboard because he needed 26 liters of intravenous electrolytes to replace the fluids he lost in that study. Cholera studies have been done for some time, and were actually recently instrumental in licensing a traveler's cholera vaccine. But these studies are very burdensome and in some cases can be fairly risky. So what's interesting about challenge studies is that they've been around for a long time. In 1796, which is widely regarded as the start of modern vaccination, Edward Jenner challenged his gardener's son, James Phipps, with smallpox. And what he did was he had noticed that cowpox was something that milkmaids were exposed to, and they therefore seemed to be protected from smallpox. And there was a long history actually of inoculation for smallpox. So he decided to use that approach to see if he could first expose Phipps to cowpox and then see if he was protected against smallpox. And this was, of course, ethically problematic on a number of levels, but really was the foundation of our modern approach to vaccination to prove that this kind of approach could work. And over the years, there were many other trials, some like Walter Reed's yellow fever experiments in Cuba, which really demonstrated important scientific advances and a lot of thought to ethical issues, although still probably wouldn't meet the standards that we have today. But also some really ethically controversial studies like the Willowbrook Pediatric Challenge studies, or unethical studies like the Guatemala STI studies, which President Obama publicly apologized for, where vulnerable participants were exposed to pathogens without their consent, without full understanding, and without protection for others who they were interacting with. Despite this long history, the first article on the ethics of controlled human infection studies was written in 2001 by Frank Miller and Christine Grady. And more recently, challenge studies have received more attention. And a big reason for that is that you may have seen that there's a lot of interest right now in using challenge studies for addressing the novel coronavirus. The idea here is that this may be a way to accelerate testing of the vaccine, which we sorely need to address the current pandemic. And many have really leapt to advocate for the use of challenge studies to accelerate vaccine testing right now against COVID-19. What I'd like to do first is take a quick step back from this advocacy to note that there was this existing ethical consensus before the pandemic about controlled human infection studies. So although ethicists who looked at this issue generally agreed that controlled human infection studies aren't different in kind from other research, in particular, they would compare these studies to, say, phase 1 studies in healthy volunteers, and note that in lots of different types of research studies, some people are exposed to risk for the benefit of others in society, not for their own benefit. And there is some level of a trade-off between that risk and benefit that we accept generally in research. So controlled human infection studies are not different in that way, but they still seem to be different, and they're still hard for the public to understand. So people noted that there's this general consensus that some diseases would be too risky for controlled human infection studies, such as HIV and Ebola. And the thought was that it was primarily because there's no curative treatment for these diseases, and the disease is what people refer to as self-limiting, where the disease would resolve. So when it's okay to do a challenge study, in other words, people thought you had to have a treatment in place to rescue participants who were infected, and you had to ensure the disease would run its course with supportive care, and people would not have serious long-term consequences. There were also several ethical considerations that were identified to apply to controlled human infection studies. And this is just a list of the different ethical considerations that people drew out. But analysis was still fairly limited. And the COVID-19 pandemic has made clear that a novel framework requires more analysis. It seems really important, particularly in this historical moment, to explicitly incorporate justice and population level concerns to make sure that a fair participant selection is addressed, and to ensure that in general, although this idea of excluding vulnerable groups makes sense and is important in many cases, it also can deny population's benefits of research in the long term. So the question about when and whether to include vulnerable groups is something that ethicists are now saying requires explicit attention. Secondly, there's also recent and more rigorous analysis of ethical concepts that were previously sort of taken for granted, things like social value, vulnerability, and risk to people outside of the research or third parties. So the challenge with controlled human infection studies then is that although they're not unique or distinct from other types of research, they do raise this constellation of difficult and unresolved issues in research ethics, including things like, what's the upper limit of risk? This is a question that ethicists have been struggling to resolve for many years and have not gotten much closer to resolving. The question of when population should be excluded as a default versus included is another challenging issue. Risks to third parties outside of research is an issue that really hadn't received a lot of attention until the mid-2000s. And finally, the right to withdraw is another important question. Although it's generally accepted participants have a right to withdraw at any time from research, that becomes more complicated when they've been infected with a pathogen that they could then spread to third parties outside of the research if they were to withdraw. So managing these issues in the context of controlled human infection studies really takes extra attention. This led me with an interdisciplinary group of challenge study researchers and ethicists and social scientists from around the world to develop a more rigorous and comprehensive ethical framework for controlled human infection studies. And I reached out to the World Health Organization when we were starting this work, and they were also interested in beginning this process. So we've been working for the past few years on developing guidance documents that could speak to the issue of controlled human infection studies in general. But more recently, we realized that it was important to rapidly repurpose this work to address COVID-19, as this is sort of the issue of the day, where this question of whether or not we should use controlled human infection studies has been raised into the public consciousness in a way that we've just never seen before. So our group wrote an article that was published in Science magazine to address the ethical criteria that we think are important. And I was also a member of a working group that developed key criteria for the ethical acceptability of COVID-19 human challenge studies for WHO. This is a summary of the issues that we laid out, and I'll go through each of these in turn. But we developed this ethical framework that laid out seven ethical considerations that should be applied, and tried to figure out what they implied for studies with that would address COVID-19. You'll note that we use SARS-CoV-2, as some others have done in previous presentations, because we're talking about deliberately exposing people to the virus that causes COVID-19. And I'll go through each of these in just a moment. But I wanted to first note that the World Health Organization criteria and the science paper, for those of you who are in the weeds on this issue, you'll notice that they're slightly different and may wonder, you know, how are these two documents supposed to relate to each other? So these are slightly different groups, and we worked to make sure that our guidance was compatible, but isn't exactly the same. The key differences are terminology. Really, we refer to controlled human infection studies in the science ethical framework, but the key criteria uses different terms. We also use the term social value in the science ethical framework, but you'll see on the left that the WHO uses the term scientific justification, which is meant to encompass the same thing. And there are 8 criteria in the WHO's key criteria. They included expert review. We just assumed that that should occur. Additionally, we thought that payment was important to address in the science ethical framework, so we also addressed that. And the WHO's key criteria document is longer and more detailed, but ours is subject to a 2,000 word limit, so there's a lot that we couldn't say. So first I'll talk about the most important criteria for research in general, but also a really critical issue for challenge studies in particular. Because of this higher risk of doing SARS-CoV-2 controlled human infection studies and the controversy around these studies, it's really important to ensure that they're worth doing and that the value or the benefit they can bring to society is rigorously evaluated. Now, in looking at value, it's important to look at the magnitude, the amount of health benefits, the distribution of those health benefits, who's going to receive them, and the likelihood that they will actually come to pass. This requires thinking about where do controlled human infection studies fit in with regard to other types of trials. How are stakeholders going to coordinate in order to use the data from controlled human infection studies? Because they really don't stand on their own. They have to be taken, the data are either feed into a vaccine development pathway or a package for licensure, but also have to be carried forward by others. It's very rare that a controlled human infection study on its own would be sufficient to say all of the world's population should receive this vaccine. Additionally, the path from controlled human infection studies to realizing health benefits can be complicated, and in particular, without equitable widespread access to proven interventions, the claim that a controlled human infection study will really make a difference has to be walked back quite a bit. Unless these interventions that are produced actually are available to people, they're not going to produce the social benefit or the amount of social benefit that they could. So there are different ideas for when social value could be realized in a controlled human infection study for the novel coronavirus. But the first that was the first out of the gate came from Mir et al., Peter Smith, and Mark Lipsitch, and their view was that controlled human infection studies could replace efficacy testing. So in the phases of vaccine testing, once the safety and immunogenicity are established of the candidate, you still have to tell whether that vaccine works. And what these 3 authors argued is that you could use controlled human infection studies to rapidly, with about 100 volunteers, test whether the vaccine works, and then maybe do some additional testing in the real world just to make sure that holds up, but rapidly make it available. And their view is this would save a couple of months. And this would have clear social value if it worked, but there are a lot of concerns to account for. First is that regulators may not accept these data. They might have a lot of concerns about generalizability if you have a non-natural way of infecting somebody, and it's just 100 young, healthy volunteers that you've tested this vaccine in, but the vaccine is meant to protect the world's population. It's also important to note that controlled human infection studies can take 4 to 12 months to launch. And although people have started to do some of the prior steps to lay the groundwork, vaccine testing has started and is speeding ahead. So Moderna, a pharmaceutical company, just announced that they have proceeded through the stage of finding immunogenicity for their vaccine trial. China recently announced some results from their trial. It's unclear how controlled human infection studies, it seems like they may have to catch up to where vaccine testing already is, and it may they take too long to set them up for them to actually catch up. So that's the first possible way these studies could make a difference. A second is to identify correlates of protection. This is the idea that you could reliably tell when people were protected against infection. And this may be something like a certain level of antibody, or it could be something else if immunity is mediated in some other way. Now, this could be useful in future vaccine studies to provide a different endpoint that you could use rather than just giving people the vaccine or a placebo vaccine and waiting to see if they became infected. You could also look for these correlates of protection. It could be used in public health responses. Some people have been talking a lot about immunity passports, and it could be useful to figure out when or if those things are a possible tool. But it will require coordination of stakeholders to use these data in order to actually make a difference. So then a third approach would be to select the most promising vaccine candidate. And this may be important because there are over 100 vaccine candidates that have been produced to date. WHO and the National Institutes of Health have narrowed it down to the 10 highest priority candidates. But it's also true that we will probably need more than one candidate to meet global demand and bring prices down. And vaccine manufacturers are already starting to manufacture at risk. So they're developing their vaccines and producing them even before they know if they will be the candidate that makes it through the gate and is actually licensed. So it is unclear here how coordination would work to make sure that a controlled human infection study that was launched would actually make a difference in behavior that would then lead to a vaccine coming to market sooner or being a better vaccine than some of the other approaches. So the final approach that I think is really important to consider is that our best laid plans may not work. And it may be that vaccines need to be tested regardless. And this could happen if physical distancing measures actually work well enough, or the epidemic wanes and field testing is unable to recruit sufficient patients. Or it could be that the first vaccine that's approved is really expensive or isn't safe enough or isn't effective in certain populations. So there are other possibilities for which a controlled human infection study could be useful if we have these quick ways to test other candidates. Finally, there are other sources of the social value even beyond what I have said. So you could gain basic scientific insights into mechanisms of infection and disease. For instance, how do people who are pre-symptomatic transmit infection? You could select the most promising treatment candidates, or you could determine how SARS-CoV-2 spreads. The challenges with all of these things, and the takeaway that I want to leave you with on social value, is that all of these potential sources of value depend on effective coordination with others and scientific and public health developments. What else is happening? So we need advanced coordination. This suggests that it makes sense to lay the groundwork now and decide whether to proceed with, and also what the primary aims would be for SARS-CoV-2 tries closer in time to launch. So the second issue, in addition to social value, is to ensure there's a reasonable risk-benefit profile. And this is difficult because you have to first identify risks, but in the current context, there's major uncertainty about risks. The infection fatality rate isn't really easy to get because of testing not being widely available. It's also true that long-term complications have not yet fully been characterized. We think that young, healthy people are at lowest risk, as some of the other panelists have mentioned, and some of the data suggests that younger people might be at relatively low risk of death and hospitalization. But these data are really still emerging. When we looked at comparing SARS-CoV-2 controlled human infection studies and other types of controlled human infection studies or phase 1 trials with healthy volunteers or other altruistic activities, we felt that it was clear that the risks are still slightly higher from doing these kinds of controlled human infection studies than other comparable activities. So this gave us pause because here what we're doing is only comparing risks of mortality and not considering long-term serious risks of morbidity. So it makes it difficult to know whether these trials could be justified. an important caveat is that a lot of the data we have are based on population level data that relies on young people pooled and including young people who have comorbidities. So what we really need are data from young people who don't have comorbidities and also a sense of what the true risk factors are that could be measured. So in order to move forward with a study like this, risks and benefits to participants should be systematically identified, evaluated, minimized, and be found to be below the upper limit. And although there are many ways that you could potentially do this that others have also discussed, like enrolling young healthy participants, monitoring them closely. Some people have talked about benefits, but I think those are all speculative since we don't really know if people have long-lasting immunity from infection in all cases. But participants could also be provided prompt treatment and compensation for research-related injury. The issue, I think, is we still don't have a consensus on the upper limit of risk, and the analogies to research or other altruistic activities suggest that this is close to or pushing that upper limit. Additionally, you also have to consider risks to third parties outside of the research. So because controlled human infection studies involve deliberately exposing participants to pathogens, they can impose risks to third parties if participants infect research personnel or their contacts, such as household members. In other studies, you might have sexual partners be infected. The study could also negatively impact the local healthcare system by using constrained resources that are needed to manage the outbreak. So we thought it's really important that risks to third parties are low, and there are ways to do this for a SARS-CoV-2 challenge study. So you could confine participants in a highly secure facility for at least 3 weeks, perhaps longer, and personal protective equipment should be made available for research personnel to avoid infecting them. But with regard to risks and benefit, this is also rapidly evolving, just like social value. And with the rapid production of data about risks, and some treatments being approved, but no cure or elimination of mortality and serious harms, it seems justified to start laying the groundwork for controlled human infection studies now, and then decide whether the risk is acceptable when they are ready to launch. We think there are several other ethical considerations, so I'll quickly run through 4 more ethical considerations. The first is context-specific stakeholder engagement. And the reason for this, although some people have sort of poo-pooed the notion of the need for engaging communities and worried about the time it would take, is that risky research can inadvertently undermine forms of broader, longer-term collaboration that sustain the production of socially valuable medical knowledge, as Alex London and Jonathan Kimmelman have argued. And they point out that major adverse events can undermine institutional support for an entire field, including gene transfer research. Although it's hard to see beyond the fog of the current pandemic, we will need research going forward. And so it's important not to do something today that would undermine research for years to come. Although these concerns about public trust are intuitively plausible, there's limited empirical evidence. But there's reason to think controlled human infection studies might be more likely to see distrust than other studies. Thank you. So we thought it's important to consider the context, to engage the public through virtual community advisory boards or the media, and gather public input in creative ways due to physical distancing. Additionally, this coordination is critically important, and even doing something like embedded social science research with the participants could also be very useful. Site selection is another important consideration. I won't discuss this at length, except to say that there are many factors to consider, and it's important to avoid interfering with the local outbreak response, but also picking communities that have already had these relationships with research and are sensitized to challenge trials. Fair participant selection is critically important as well. Here, I'll leave you with a take-home that populations should not be chosen due to vulnerability or privilege, and that they should have the capacity to provide voluntary informed consent, but that minimizing risk is one of the most important considerations. Robust informed consent is really important, but I think it sort of comes after the justification for the study has been established, and there are evidence-based approaches to enhance understanding, including things like tests of comprehension, which are regularly done in controlled human infection studies. This would also have to be regularly updated, and key elements are the things that you would focus on understanding and making sure participants understand them. Finally, payments, which I won't have time to talk about, but I'd be happy to answer more questions. In general, I think it's important that participants are compensated for time and expenses to avoid exploiting them. People should not be subsidizing research or unable to participate because they have other duties and they can't afford to pay for their participation. However, if large amounts of money, particularly in the current context of economic deprivation, leads people to withhold information about their eligibility, that could be problematic. And that could be addressed by designing objective tests to verify eligibility and explaining to people the importance of eligibility criteria for scientific and safety reasons. Now, there's this high level of interest in participating. Some of you may have seen the One Day Sooner group, which has actually now over 28,000 volunteers who've signed up for COVID-19 challenge trials. So any payment beyond compensation may not be necessary given this high level of interest, although there are lots of open questions about who these volunteers are and what they understand, and I'm actually participating in a survey of these volunteers to find that out. So in conclusion, it's worth investing in controlled human infection studies with the novel coronavirus now to ensure they will be ready to deploy if they are needed and if they are justified. The potential social value and risk of these studies is very dynamic. So it will be important to develop a robust process to determine whether they are justified before launch, and that process should be immune to political pressure so that the people who are involved are able to say no if that makes sense, or yes if it does. Finally, even if these studies become justifiable, I have briefly described other considerations that should also be addressed to ensure that these studies are conducted ethically and that the institutions conducting them are worthy of the public's trust. Thank you. I'll just leave you with some acknowledgements of our incredible team and see if there are any questions.
Thank you very much, Seema, for a very interesting and provocative talk. So Seema, how is it possible to quantify the actual risk to a patient before, one of these exposure trials is actually initiated? It sounds like, you may not know what you're going to get. It's like.
Yeah. So certainly with the current data,
I mean, I guess the better question is how do IRB interpret, for instance, the request to run such tests in the absence of data on potential, adverse effects on the participants?
Great. So I think that, IRBs are accustomed to dealing with whatever data are available, whatever the best data are. And with emerging infectious diseases, that's harder because the data are just from a very short time period. So you really can't have data about long-term complications. At the same time, research on emerging infectious diseases is so important that shouldn't stop conducting this research. And we do live with uncertainty in other cases. So I think it really is about looking at the best data where there are some modelers who are developing data to try and see, what would be the risks to young, healthy people. And there are obvious limitations of modeling, but you could use those data, as we have tried to do, and come up with a number about what the level of risk is likely to be. So, we've found this number of about 3 and a risk of mortality of about 3 in 10,000. So if that is right, these studies may be acceptable, but it's hard to know if that's right now. And as more data emerge from these, as the infection spreads around the world, it may be in the next few months, there's a better sense of who's at risk from mortality and also better knowledge about why some young, healthy people are still having these serious complications. And that could give IRBs more confidence about letting these trials go forward.
Andrea?
Yeah.
Oh, sorry. Could I ask just a
quick
couple of questions?
Sure.
You mentioned the One Day Sooner project, and I was just wondering, is that something that's actually going to be implemented, or are they just seeking volunteers in case it were implemented? And then the second question I have is, are there any currently ongoing controlled human infection studies for any other diseases?
So let me take your first question first. There are many ongoing controlled human infection studies for diseases. So they're commonly done for malaria, influenza, cholera, typhoid, and they've been instrumental in licensing typhoid and cholera vaccines in recent years. So they are powerful tools. I was the chair of a panel for NIH on the ethics of Zika virus challenge studies, and that during the epidemic, we felt there wasn't enough known about risks and also how long people could transmit the infection to third parties. And there were lots of other studies ongoing, but that epidemic sort of flamed out where there's still small outbreaks that seem to be going on, but those outbreaks are not things that are predictable enough to launch vaccine trials. So vaccine trials are stalled at the moment that people think Zika is a threat that hasn't gone away. And Zika virus controlled human infection studies are now being planned as the only way to move vaccine testing forward. So that's probably the one case of a truly emerging infectious disease where we still don't know a ton about it, but it is one where the social value of doing that trial is pretty large and could justify exposing people to risk. And we've also learned enough to protect participants. With regard to the One Day Sooner group, it's an independent advocacy group that has created this website. It's unclear to me yet whether this website will be used to feed into challenge studies that are being planned, but there are 3 companies that are currently considering doing controlled human infection studies of the novel coronavirus. And even at the governmental level, there are governments who are seriously considering this. In general, I think the current consensus is people are waiting to see whether there's a treatment or whether it's clear that vaccine trials wouldn't work. And if those 2 things come to pass, then I think that we will probably see controlled human infection studies move forward with the novel coronavirus.
Thank you.